Exit seminar. Flowers. Research and technology product group. So this is obviously a big day, an important day, also kind of a bittersweet moment because this is the moment to see Minty move on. But it's been an incredible experience to have Jasen in the lab. I think of Jasen as someone who really embodies, sort of the true pandemic Asian student. Jasen arrived here fall quarter 2019. He came from UC Santa Barbara with a major in biology and statistics. And at that time, everything was looking nice. And then 6 months later, what happened? It was kind of a challenging time, and I was reflecting on all the things that we've gone through together, and it really is amazing. I feel extremely proud of final product that Jasen has been able to produce. And it was really challenging for Jasen because his idea was the interactions between orchids and perfume flowers, which obviously require going to the field, travel internationally. And I think two field trips, I think, were canceled. And it wasn't really until 2022 when Jasen was able to finally go to the field. And that's when I took this picture of him. We went to the field together, southwestern Costa Rica. And this picture really captures a lot, I think. It captures, well, for one, Jasen is extremely happy. He's holding a stash of Gongora orchids, which is one of the subjects of the final chapter that he's working on. And it also captures the habitat where these orchids live, exactly on those rivers, and captures the sort of the really complex diversity of these organisms. And it also captures, I think, the sort of pivotal moment in Jasen's dissertational research. This is the moment where I think Jasen really started to flourish in his research. It was really hard to not being able to go to the field. And after this field trip, I think Jasen really expanded his horizons and started to do a lot of really interesting research, which combined field work, international collaborations, analyzing molecular phylogenies and chemical data sets. I'm not going to go into the details. He's going to tell you all about it. And the final product is really amazing. Jasen is thinking in very, very broad scales, macroevolution, but also thinking in the details of natural history and species interactions that really matter for the evolution of these organisms. Leave it there, and thank you all for coming.
Hey everyone, thank you Santiago for that lovely introduction, and thank you all for attending my exit seminar. I'm really excited to share some of the work that I've been doing for the last 6 years. So I'm going to start with some of the motivation for my work, and it starts from this observation that flowering plants are extremely diverse. So there are around 300,000 extant species compared to around 30,000 total non-flowering land plants. And it's coevolution with animal pollinators that are mediated by these flowers, these specialized reproductive organs that have thought to driven the diversification of this group. And we know that now that animal pollination spurs plant diversification. Around 90% of flowering plant species require the services of animal pollinators for successful reproduction. And as these animal pollinators span great taxonomic breadth, from birds to moths to bees, this can generate really immense opportunity for these pollinators to exert different selective pressures on these flowers. For example, we might imagine that this hummingbird is going to want different things from the flower than this bee. Birds are primarily visual, insects often rely on chemical cues, and the rewards that they're after are often different. So 2 mechanisms of prezygotic isolation, or sort of selection from different pollinators, can result in the evolution of these different populations of plants. And these 2 mechanisms are broadly boiled down to mechanical and ecological isolation. Mechanical isolation is due to morphological differences that prevent pollen transfer by the same pollinators. So here we have 2 species of columbine that vary with respect to the lengths of their nectar spurs, these tubular extensions of the flowers with nectar rewards as the base, as well as their flowering the positions of their flowers. The one on the left is sort of dangling downwards, the one on the right is upright. And only pollinators with proper foraging positions and feeding apparatuses that roughly match spur lengths will have good morphological fit with the reproductive organs of each flower, facilitating the reproductive success. For example, this hummingbird is foraging from this bottom position, and you can see that its head is contacting the anthers of the flowers, whereas this hawk moth has a really long tongue and it's foraging from this upright angle, thus encountering the pollen-bearing organs of this flower. Ecological isolation, on the other hand, is isolation due to the use of signals that target different sensory biases exhibited by different pollinators. So here we have two species of East Asian mitella that grow in the same regions, but they actually are able to attract different pollinators because they produce different scents, these different compounds in their floral volatiles that attract these different species of fungus gnats. So for my dissertation research, I was interested in how pollinators influence the evolution of isolating mechanisms in plants and focused on a particularly charismatic group of animals, those being the euglossian bees. So these are a really fantastic group of bees. A lot of them are really shiny. They look almost like flies, but some of them are pretty big. They look like bumblebees. They're hairy. There are around 200 species found in the neotropics, so from Mexico to Argentina. But really what unites them and makes them interesting to study in the context of pollination is this really unique behavior exhibited by males, which is perfume collection. So males exhibit this specialized hind leg morphology. You can see it looks almost like a pouch. And this pouch is used to store compounds for courtship. So these males will essentially go through the environment and collect different compounds to use in their courtship displays. And this acts as a pheromone analog. So instead of most other insects, which internally synthesize their own pheromones, these bees are collecting them from the environmental source, from the environment, including from a lot of different flowers. So this is kind of what perfume collection looks like. Here you can see this bee, he's investigating this flower, and now he's landed on the scent-producing region of the flower. You can see he's kind of brushing against this region with his forelegs. So there are these specialized brush-like components on his forelegs. And in a minute, he's gonna fly off. And he's completely entranced by this process. I'm like standing maybe a foot away and he's not perturbed. You can see that he's now rubbing his front legs against his back legs, basically putting the compounds into that pouch. And we have over 1,000 species of plants in neotropics that are exclusively pollinated by male euglossian bees. And I'm going to be referring to these as perfume flowers for the rest of the talk. And I really wanted to emphasize that this is truly a unique pollination system. So as opposed to most systems where floral scent is sort of advertising the presence of another reward, so basically a flower is smelling nice because it's telling its pollinators that it has nectar. And here in these systems, this scent is both the attractant and the reward. So scent is acting as the long distance reward, and it's also exactly what the bees are visiting the flowers for. So critically, there are many examples of the isolating mechanisms that I described earlier. So first, there's an abundance of mechanical isolating barriers in perfume flowers. So a lot of these communities where these orchid bees are found in are pretty diverse. They often contain over 40 species of bees. In some parts of Ecuador, there are up to 66 species. And this generates the opportunity for selection to put your pollen on different parts of the bees. Essentially, you want to avoid hybridization that might be caused if you're putting your pollen grains on the same area of a bee as a different flower. So these beautiful pictures by Adam Karamans are showing a bunch of different orchid bee species, and these yellow structures, like here and here, are showing these different pollen packages placed by different species of orchids. And you can see that they're on really different parts of the bees. So this one, they're on the thorax, this one's on the abdomen, this one's on the antennae. And these mechanisms are also often extremely precise. If you look at this first bee, you can actually see that there are 3 separate packages of these pollen placed on his body. So essentially, he visited 3 different flowers, and all 3 of those flowers placed those pollen packages at the exact same spot. And this diversity of placement strategies is really testament to the abundance of mechanisms mediated by morphological differences. Or in other words, perfume flowers are really strange. So this is an example of one. This is Coryanthes, also known as a bucket orchid. And it truly, like, if you look at this, it's kind of difficult to parse what part of this corresponds to what part of a flower. And it truly takes bees on a whimsical journey. So unfortunately, the sound is not going to work, but I'm going to try to narrate. So here is a bee. He's collecting scent from the scent-producing regions of the flower. Really strange. You can see the bucket morphology described earlier. And this bucket is actually filled with fluid that the flower produces from a different region on itself. And you can see that it's kind of a slippery region. So he slips into the bucket and now his wings are wet, so he can't immediately fly away. If you were watching with sound, there'd be dramatic music happening. And now his only method of escape is through this one narrow passage, which he has difficulty navigating because he's wet and it's sort of a slippery environment. But what's really cool here is now that as he's exiting, that over here is the anther cap. So basically right underneath is where all the pollen structures are. And he's like, this flower is really gripping him tightly. So it takes a while for him to get out. Sometimes it can take upwards of an hour. But you can see now that the flower has basically glued these pollen packages onto him, and now he's escaping. So it's wild that a pollination strategy like this works, and especially if you think about the fact that for successful reproduction to happen, this bee needs to do this twice. So clearly he's very happy with what he's getting, or he's very attracted to the rewards. Additionally, we have species like Catasetum, and they really forcefully catapult their pollen structures onto their pollinators. So pay attention to this bee. This happens really quickly. You can see that that orchid places this massive pollen package onto the body of that bee. And here we can watch it in slower motion. So again, focus on this individual. And you can kind of imagine that if you're that size, that might be really forceful. But again, this bee has to do this twice, or yeah. And these perfume flowers are strange, but I'm not the first to notice this. In fact, Darwin himself wrote about these orchids. He was really fascinated by them. He didn't actually observe them in the wild, so he never actually observed the pollination. But in the greenhouse, he sort of manipulated them and figured out that, these plants must have had really complex mechanisms to facilitate pollination. So for ecological isolation, scent is extremely important. So every bee species sort of exhibits its own species-specific scent preference. Essentially, every male is collecting a different set of compounds to produce his perfume bouquet that he presents during courtship. So from the plant's perspective, essentially slight changes in floral scent can result in reproductive isolation because you're attracting different sets of pollinators. So here's kind of a little cartoon model of how this works. Here we have an ancestral population of some perfume flower that's producing these scents and is attracting these two species of bees, this purple and this green bee. And we can imagine as time moves forward, one of these populations still produces the same scent and is attracting the same species of bees. But if along this branch we see some divergence in the chemistry that's produced, we might see that this population is going to attract different species of bees, this orange and this yellow one, thus reducing gene flow between these populations. And this suggests that speciation can be facilitated through relatively simple changes in floral chemistry. And actually at the macroevolutionary level, we see that male euglossian pollination is actually associated with faster net diversification rates in orchids, potentially because of how easy it is to have this reproductive isolation. But despite this work, understanding patterns of trait variation is less well understood in these groups of plants. So my work seeks to address these gaps by understanding trait evolution at several scales. And my research is grounded within this phylogenetic framework that you're going to be seeing a lot in my talk. So here is a phylogeny of this group of orchids that I'm interested in, where turquoise colors represent non-perfume flowers and pink colors represent perfume flowers. And you can see that there are five origins of male euglossian pollination within this group. which offers a really great opportunity to test hypotheses about how these bees influence trait evolution using these independent evolutionary replicants. Which gets to my first chapter, which is elucidating the role of male euglossine pollination on floral morphological evolution. Or in other words, how do we make sense of the abundance of mechanical isolating barriers in the perfume flowers? For the second chapter, I'm going to focus only on these perfume flowers and try to understand evolutionary patterns of floral volatiles within these groups. So essentially, trying to understand how perfume flowers smell and the patterns of volatile evolution. And finally, I'm going to be focusing on a single one of these pink branches, exhibiting variation in chemistry to try to understand some of the genetic mechanisms that underlie differentiation of this trait. So together, my research aims to understand how euglossine bees have influenced floral trait evolution in a truly iconic radiation of plants that have inspired biologists and horticulturists since Darwin's time. So jumping into my first chapter, elucidating the role of euglossine pollination on floral morphological evolution. So again, back to my motivation, perfume flowers are strange. Again, we see these really complex pollination mechanisms, including going through this bucket filled with liquid and this forceful pollination or forceful placement of pollen structures. But the question is now, are they stranger than expectations? So as many of you might know, orchids are extremely diverse. They have immense levels of morphological variation. So the question is, do perfume flowers exhibit elevated rates of evolution compared to background rates within this super diverse group? For example, shown here, there are a few different orchids. These look really different from each other, but they're only around 10 to 15 million years diverged. So to test these hypotheses, we need these three components. We need a phylogeny, we need our data, and we need some model to test the hypothesis. So to start with the phylogeny, I used this time-calibrated phylogeny of the Cymbididae, which is the group that I showed earlier with those 5 independent origins. And I pruned it down to my focal species. So this is an extremely diverse group. They're a group of orchids that arrived in the tropical Americas around 25 million years ago and really rapidly diversified into 3,000 extant species. Next, we need our data. So for my data, I collected a lot from these beautiful Lancaster digital composite plates, or essentially digitized botanical descriptions, where they dissected these flowers and took a lot of these standardized photographs. A lot of this work was done by Diego Bogarin, Franco Pupulin, Adam Karamans, and Eugenio Restrepo. In addition, I took some of these photographs myself from private collections. And we got around 140 species, which is not a huge amount in terms of the total diversity in this group, but it covers 70 genera, or around 50% of this sort of broad scale of taxonomic organization within the group. And to collect the data, we took 9 linear measurements of the petals and sepals shown here that sort of capture these patterns of width and length. In addition, on the lateral views of the reproductive structure, we set a series of curves to capture overall shape variation. And a lot of this was done by my wonderful undergraduate mentee, Yang Xie. So jumping into the results of what the data looks like, this is a principal components analysis of the floral morphospace for just the petals and the sepals. And here, every point represents a different species. Points that are closer together are more morphologically similar. And to kind of walk you through the extremes of this variation, along this first axis of variation, on one side, we have species that have these really stout, rounded petals and sepals. And on the other end, we have species like this Brassia or the spider orchid that have really elongated petals and sepals. So for morphologists here, this might seem pretty familiar. This is sort of an elongation ratio, which is pretty common in a lot of data sets. And on the second axis, on one end, we have this species of Telepogon, which has a really long sepal, or long petal and short sepals. And on the other end, we have this Gongora with really large sepals and tiny petals. So this second axis of variation is sort of capturing the relative lengths of the petals to the sepals. And finally, at the very origin, so sort of the average phenotype within this group, you have this Cameridium, which has sort of intermediate levels of both. The petals and the sepals are roughly the same length. There's sort of this intermediate elongation or elongated appearance. They're not super elongated, they're not super stout. Next, if we look at the reproductive structure, so on one end, we have Coryanthes, or the bucket orchid that I showed earlier. Kind of makes sense that this would be on one end of one extreme of the variation because it's such a strange flower, has this really pouched appearance. And on the other end, we have species like this Cyrtocylum with this really reflexed reproductive structure. So this first axis is sort of capturing this pouched to really outward appearance. And on the second axis, on one end, we have species like this Cagiliella, and on the other end, we have this Ornithocephalus. So the second axis of variation is sort of capturing the relative lengths of these two components of the reproductive structure. Here, the first part is really short compared to the second part. And here, this component of the reproductive structure is really long compared to the second. And if we look at the orchid that's sort of at the very center of this diversity, so the average phenotype, it's actually the same species over here. So it's overall very average orchid. And again, you can kind of see that it's intermediate in all of these traits. So this kind of gives us a sense of what the overall diversity looks like. But if we color these orchids by the pollination mode, we can here with the turquoise points representing non-perfume orchids and the pink points corresponding to pink or to perfume orchids, we can see that actually both groups contain equivalent amounts of morphological variation, or really similar amounts of morphological variation. So the question is now, was this accumulated at a more rapid rate, given that there are that these origins of perfume flowers occurred relatively recently? And that's where the model has to come in. So to test this hypothesis, I used a multiple state-specific rate of continuous character evolution, also known as muskrat. And I implemented this in RevBase. So with these three pieces, I ran the model and a week later got this output. So this is a lot to look at, but I'm gonna walk you through it. So essentially what you're seeing here, these two phylogenies are the same. The one on the left is corresponding to the petal and sepal data set, and the one on the right is corresponding to the reproductive structure data set. And the colors correspond to the rates of evolution in those two sets of traits, with yellow or these lighter colors corresponding to faster rates and these darker purple colors corresponding to slower rates. And in the center, we have these species coded by their pollination mode. And the first thing is that you can see that there's a lot of rate variation across the tree, lots of different shades of yellows and purples. But we also see that there's elevated rates in perfume flower clades. So if we look at this group on the petal and sepal data set, this corresponds to the group that contains the bucket orchid. So it's one of the large radiations of perfume flowers. And in this group on the right for the reproductive structure, this corresponds to the group containing catasetum, the one that really forcefully places its pollinarium. So we see that perfume flowers exhibit the most rapid rates in both groups. And in addition, if we sort of plot the posterior estimates of the rates associated with each pollination mode, we see that perfume flowers evolve faster than non-perfume flowers at a rate over 5 times faster in the petal and sepal data set and around twice as fast in the reproductive structure data set. So in conclusion for this section, we see that perfume flowers exhibit equivalent amounts of diversity as all other pollination mechanisms in this clade. And this was achieved via elevated rates of evolution. And these results sort of provide some new insights into the capacity for pollinators to exhibit selective pressures on flowers, providing the engine for trait diversification in some of the world's richest floristic hotspots. So moving on to chapter 2, talking about scent. Again, scent, as I mentioned earlier, scent divergence drives speciation in perfume flowers. So we know that scent plays this really important role in the process of speciation. But few studies have actually addressed these sort of large patterns of variation across perfume flowers. So for this chapter, the two main questions I tried to address were, one, what are the major axes of scent variation across this pollination mode? And two, what is the tempo and mode of scent evolution? Or in other words, first question is asking what compounds are present, and the second question is asking, do closely related species exhibit similar scent profiles? And for this project, I collaborated with Paulo Milet Pinheiro and Carlos Nunes, who had this wonderful data set of 178 species with scent data spanning over 50 years of published research. And most of this data set is from the orchids, but they're actually members of these other 5 families as well. Araceae, including peace lilies, which are a common houseplant, and even some members of Solanaceae, which is the group that contains like tomatoes and potatoes. So to get a kind of general sense of how scent data are collected, essentially you bag a flower in an oven bag, the same one you would roast a turkey in, and then you let that scent accumulate. And afterwards, you draw the air through this adsorbent tube that contains these polymer materials that gather scent on them. You then elute these compounds out of that polymer with some sort of solvent, and you run it on our gas chromatography mass spectrometry instrument. And the output is essentially a fingerprint of what the scent profile looks like. So here, every peak corresponds to a different compound, and the height of each peak corresponds to its relative abundance in that mixture. So jumping into these two questions, one, what are the major axes of scent variation? So again, looking at broad patterns, this is a type of plot where every point represents a different species, where points that are more close to each other are more chemically similar. And each point's color here represents the dominant biosynthetic pathway that's represented in its scent profile. So we have a lot of species that are terpene dominated in dark blue, and a lot of species that are aromatic dominated in light pink, with relatively few individuals that are sort of intermediate or exhibit other biosynthetic pathways. So to get a sense of what some of these compounds look like, terpene compounds are often these 10 to 15 carbon structures, including things like limonene, which is the dominant scent in citrus, and myrcene, which is the dominant scent in hops. And subtle chemical modifications can result in really different smelling compounds. So for example, if you add an alcohol group to this myrcene, you actually get linalool, which is the dominant scent in lavender. Aromatic compounds, on the other hand, are products of this different biosynthetic pathway and include things like eugenol, which is the dominant scent in cloves, and methyl salicylate, which is the dominant scent in wintergreen. And Peter is going to pass around some compounds if you want to smell them. Don't puff too hard and just be smart. So yeah, just to get a sense of what some of these different species smell like, we have species like Gongra claviodora, which has a scent profile that's actually completely composed of eugenol, or this clove compound, and its specific epithet, claviodora, reflects this. In the terpene-dominated plants, on one end, we have species like Catasetum purum, which has mostly these linear monoterpenes. And on the other end, we have species like this Coryanthes flava, one of the bucket orchids, that has mostly these ringed compounds. And in fact, this second axis of variation is actually mostly explained by this linear to ringed compound variation. So this is the exact same plot, but now the second axis of variation is sized by the proportion of that scent profile that's dominated by the single compound cineol, which Peter is going to pass around. Cineol is what's known as a generalized attractant, and it's also known as eucalyptol, which is the dominant compound in eucalyptus leaf volatile. So sort of that really minty, strong smell that you might associate with cough drops. And what I mean by generalized attractant, if you put out pure cineol, you actually attract a large assemblage of different bees. So this was cineol put on a piece of filter paper inside a tea strainer. And within minutes, these 3 or 4 different species of bees have been attracted. You have these beautiful Euphrasia pulchra that have these beautiful shiny eyes and shiny abdomen, in addition to 3 different colored species of Euglossa. So what's kind of interesting is that these major axes of variation are not taxonomically structured. So now it's the same plot, but now it's colored by the family in which the plant is from. And we can see that 3 out of the 4 families with more than one representative span this entire chemospace. So essentially we see that orchids are everywhere in blue, but also some of these other groups, for example, the Eraceae in this sort of greenish camo color, are also spread throughout. These pink members corresponding to Solanaceae are also spread throughout this chemospace. And if we look at some of these situations where we have 2 species next to each other, over here we have these 2 species, a Stanhopea, which is an orchid, and the Solanum, which are the most chemically similar plants. And if we look over here, we see that these 2 species correspond to Anthurium and Aroid, and this other Stanhopea member. So these 2 members from the same genus are actually quite distant, and they're really close in chemical space to a really distantly related species. So this suggests that there's really widespread convergence of broad chemical phenotypes, which leads to the second question of what is the tempo and mode of scent evolution. So to test this, I focused on these two different clades, the Stanhopeanae and the Catasedae. So this one contains the bucket orchid, this one contains the Catasedum. And these are two of the largest radiations of perfume flowers within the orchids. And they have a similar age of around 15 million years and a similar history of chemical ecology research in the literature. So I did what's called a disparity through time analysis. So just to walk you through what the results of a run might look like, the x-axis here represents relative time, with 1 corresponding to the tips of the phylogeny or the present day, and 0 corresponding to the root of the tree. And shown here in gray are the results of 1,000 simulations of the data using a Brownian motion process to create sort of a null distribution. And next, we plot our data as a solid line. So if our data looks like this, where it's below this dotted line or below this null distribution, it suggests that we have really strong phylogenetic signal where each clade in here exhibits a really distinctive chemical profile, or that this trait exhibits a really highly constrained structure. And if on the other hand, we see some pattern that looks more like this, where it is above the dotted line, we have evidence that there's greater disparity than by chance, with rapid filling and sharing of trait space. And if we look at our data, we see that in both cases, it's really solidly above this null distribution. Scent evolution exceeds this Brownian motion model in both clades. So it suggests that both of these clades exhibit really rapid evolution of scent and sharing of trait space throughout their evolutionary histories. So conclusions from this section, scent variation is primarily structured by the dominant biosynthetic pathways present within the perfume blend. And scent variation is also driven by terpenoid identity. And finally, scent evolves rapidly consistent with its role in the process of speciation. And you want to read more, this was published recently in Evolution, and we made it to the cover because of Thomas's beautiful. So now that we've found this pattern of pervasive chemical convergence and really rapid evolution, the final project was to characterize the mechanistic basis of scent production. So this builds a lot off of the work done by a previous grad student in the lab, Fernanda Guizar, where she characterized cryptic chemotypes of Gongora orchids in Costa Rica. So they're found in the southwest portion in around La Gamba Field Station. And what I mean by cryptic chemotypes, so I'll mostly be talking about these two chemotypes, the A and the M. As you can see, all of these individuals correspond to the A, all of these individuals correspond to the M. And what you can kind of see is that there's a lot more variation within each of these chemotypes, at least visually, than between. You can't really tell them apart just by looking at them. But they actually smell really different. So the A chemotype smells kind of bad. It's A for aromatic, and it's dominated by this sort of fishy or eggy smell. And the M chemotype is monoterpene dominated with this sort of pleasant medicinal piney scent. And they actually separate out very nicely in chemical space. So you can see here the M chemotype in green and this dark orange A chemotype over here are pretty distinct in this chemical space. And because of their divergence in scent, they attract different species or different suites of euglossine bee pollinators and are in the early stages of, presumably in the early stages of speciation. Additionally, there's really low genetic differentiation. So the A clade is actually nested within the M clade. And Fernanda found that overall across the genome, FST values are pretty low between these two populations. So my work was focusing on trying to understand the transcriptomic basis, or the gene expression that might underlie these shifts between biosynthetic pathways. So in other words, how do we get from this sort of blue region so rapidly to this pink region? And this work was helped a lot by Jasen Liu, another one of my amazing undergraduate mentees who did a lot of work in helping prep some of these samples for sequencing and in doing some preliminary analyses. So just to jump really quickly through these results, we looked at the A versus M, and we found that these differentially expressed genes are enriched for terms associated with aromatic synthesis. So that's a huge sigh of relief, kind of makes sense that sort of the biosynthesis genes are really strongly differentially expressed between these two chemotypes that differ with respect to smell. Shown here is a heat map of gene expression between the two chemotypes, where every column represents a different individual, and every line corresponds, or every row corresponds to a different gene. And we look at the topmost differentially expressed gene, this G2312. This gene is expressed over 900 times higher in A than in M, and it is potentially this O-methyltransferase. These genes are often responsible for functional modifications that create different aromatic compounds from precursors. So for example, in this plant, the species of plant, Silene latifolia, its chemical profile is mostly dominated by this veratrol compound. And this compound is actually mostly created, or the final step of the biosynthesis pathway is this OMT that catalyzes this alcohol group into this methoxy group. Kind of interestingly, if we look at the scents that are expressed in the A chemotype, both estragol and methylmethoxycinnamate have these methoxy groups. So this is potentially a gene that might be responsible for catalyzing this final step in their biosynthesis. But that kind of gets to a question of where are the precursors coming from? So to answer that question, we zoomed out on this phylogeny and looked at this other chemotype, this X chemotype, that also has a monoterpene dominated scent. It's mostly cineal, so that eucalyptus compound that you might have smelled. And we focused on this contrast between the X and the M chemotypes to try to understand if the M chemotype is sort of predisposed to aromatic production. So again, we looked at gene expression differences between these two chemotypes. And we found that a lot of the genes are enriched for monoterpene biosynthesis process, which kind of makes sense. They're two chemotypes that produce different monoterpenes. But what's really interesting is that if you look at the most differentially expressed gene, this G12713, this gene is expressed on average 64 times higher in M than in X. And it's actually a potential cinnamoyl coenzyme A reductase, which is an enzyme that's not involved in terpene synthesis. It's involved in aromatic biosynthesis. And what it does is that it catalyzes the formation of aromatic aldehydes, which are these common precursors to other aromatic compounds. So these CCRs, these cinnamoyl coenzyme reductases, sort of responsible for catalyzing the production of these precursors that are really important for production of these volatiles. So essentially, M might be predisposed for biosynthesis of aromatic compounds despite not producing any in its floral scent. So finally, for the conclusions of this section, here we have sort of a cartoon phylogeny showing the relationships between these groups. At the base of the tree, we have a probable terpene-emitting ancestor. Along the branch leading to the M and A clades, we see that CCR enzyme is expressed in flowers to generate aromatic precursors. And then along the branch to A, we just have gain of expression of this OMT that catalyzes precursors into these volatiles. So in other words, contingency or this evolutionary history really facilitates the ease of switching between biosynthetic pathways. And these results provide a framework for understanding macroevolutionary shifts in scent and provide sort of the foundation for future functional genetic validation that we would need to really prove that these genes are responsible. So taken together, my research addresses how male euglossine bee pollinators have influenced floral evolution at multiple scales. At the really broad scale, we see that euglossine bees facilitated the rapid morphological diversification of perfume flowers, leading to their abundance of mechanical isolating mechanisms. We see that perfume flowers exhibit repeated evolution of aromatic and terpene-dominated scent profiles with really rapid switching between these two phenotypes. Finally, we see that shifts between biosynthetic pathways may be facilitated by co-opting existing machinery, or this element of contingency. And together, my research shows how pollinators influence floral trait evolution at macroevolutionary scales, along with this sort of mechanistic basis underlying. So with that, I'd like to thank a bunch of people. This is sort of the short acknowledgements. I'd like to thank the Ramirez Lab, and I'd like to thank the Wainwright Lab and the Bay Lab for providing a really nice support system for doing a lot of analyses that are outside the realm of what we normally do in the Ramirez Lab. I'd like to thank the Eve front office for helping me with everything. Thank you so much, Sherry and Debbie and Joe and Shelly. I'd like to thank my collaborators, so many collaborators to thank. They've really helped me develop as a scientist and helped me so much with thinking about how I do my work. I'd like to thank my funding sources. I'd like to thank these different places where I've done my work. So at Lagamba Field Station, all the staff there who have helped provide a really supportive system for me to do my field work. I'd like to thank all the folks at Lancaster Botanical Garden for providing lab space and a really fun environment for me to work with orchids, like an entire institute of orchid researchers. I'd like to thank the UC Davis Botanical Conservatory for allowing me to collect some specimens from them, in addition to Andy's orchids. Yeah, and I'd like to thank my qualifying exam committee, Phil Ward, Michael Torelli, Peter Wainwright, Rachel Bain, Philip Zerby, in addition to my dissertation committee, Santiago, Peter and Kathleen. Colin and Denise did some illustrations that I used a lot in my presentation. And these folks contributed some of the photographs that I used in my analyses. And finally, I would like to give another big thanks to my undergraduate mentees who helped a lot with my work, Nelly Batista, Yang Xie, and Dasit Mai. And this would normally be the part where I show like the different orchids. Unfortunately, none of them were in bloom today in the conservatory. But actually, if you walk into the conservatory, just in this first part, this first like hallway, a lot of these hanging plants are perfume flowers. They're Stanthopias and Gongras. You should check it out. They have a lot of cool plants. Thank you, and I'll take some questions.
How did geographic isolation play into all this?
Oh, that's a good question. So the question is, how did geographic isolation play into all this? The Andes, the orogeny of the Andes probably did facilitate a lot of the evolution of these plants. In the phylogeny that I used, one of the key points of that study was that the Andes really facilitated the evolution of the Cymbidiae group, and that includes, like most of the perfume flowers that I'm talking about are lowland species, but there are quite a few that grow in the foothills and in the highlands. So that probably generated some, not edaphic shifts necessarily, but sort of these abiotic factors that might have promoted these plants to grow in different environments. If that makes sense. Yeah.
So the results from your chapter 2, I think it was, about the different chemical makeup of the scents reminded me a lot of the plant herbivore literature, where there's this diversity of secondary compounds in leaves. But my memory of that literature is that it is fairly constrained to family and phylogenetically conserved. And I'm wondering if my memory is wrong about the plant, leaf chemistry literature, or if there's something different about floral scents than secondary chemistry in leaves.
Yeah. So the question is, how might evolution be working in floral volatiles versus in leaf volatiles?
Sure.
I think, so there's another part of Fernanda's dissertation that was about sort of how the scent evolution might be evolving in response to florovores. So there's that potential aspect, but just kind of ignoring it. I think that for scent, because there's, scent is often produced from these common pathways that are sort of, every plant is able to express. So I think there's a lot less constraint in that regard. A lot of the leaf volatiles are these really like high molecular weight compounds that are from really specialized pathways. So I think that is sort of a mechanism in which there's less constraint potentially in the floral volatile side of things. If that makes sense.
I just want to add one thing, which is that I think there was an even easier element to the question I asked you, which you answered unintentionally, which is that some of the chemicals I'm thinking of are not necessarily volatiles. Like, I don't know if phenylglycosides are volatiles or not. So that might be part of the difference too. I don't know. Is phenylglycoside a volatile? That wasn't necessarily, yeah, anyway. I'll let other people answer.
Julia?
Yeah, I'm wondering how the female bee choice might factor into this, and whether there's some sort of like, if you have a rare scent, does that make you more attractive as a male with perfume, and how that might relate to the diversification rates?
Yeah. So so far, I think we only know of like one compound that is sort of the driver of differentiation between species of bees. So this is on the bee side of things. I don't think that compound has ever been found in perfume flowers. That compound might be fungal in origin. A lot of what the perfume flowers do is produce these really abundant floral volatiles, but just in copious quantities. So I think there's less of an influence of the perfume flowers on the orchid bees themselves than the other direction. If that kind of makes sense. Rachel.
Thanks, Jason. That was a great talk. So I was curious about the evolution of the perfume strategy, like the perfume pollination strategy. It looked like there weren't very many reversals or really any reversals to other pollination syndromes or strategies within those clades. And so I was curious if that might be a real pattern, and if so, like, what do you think is driving that?
Yeah, that's an interesting question. So we do see one reversal here. What's interesting is that in the Catasedae and the Sanhopeanae, sort of these really diverse groups, there haven't been any reversals. So I wonder if there's some sort of like progression of specialization that's required. This group, unfortunately, we don't have that much resolution with respect to like what pollinates most of the members of the group. It's super diverse, but we don't have that much pollination data. So that was a very long way of saying it depends. But it seems like some groups of orchids have these really amazing diversification following perfume flower evolution, but in some cases we do have these reversals.
Yeah, aren't there some non-fragrant gongoras?
Oh, they're non-fragrant, but they're still pollinated by male euglossian bees. What the reward in those cases might be semi-volatile, so they're like higher molecular weight compounds that don't necessarily smell to our noses, but the bees are able to detect.
Chapter 1, when you were comparing the rate of evolution in the sterile floral organs versus the reproductive organs, and you saw this kind of large difference between those two sets. One was like 5 times and the other one. What kind of selection pressures are acting on those morphological features, and why might you see quicker evolution in the,
My impression is that the sepals and the petals might just be because there's lack of selection to like create sort of a phenotype that a pollinator would recognize as a flower. For example, if you looked at the bucket orchid, the petals and the sepals, they're kind of just there. They're not really contributing anything to the pollination mechanism itself. So my impression is because the reproductive structure, those are really responsible for the actual interaction of the pollinator's body to facilitate pollination, there might be a little bit more constraint, which is why there's a little, the differences are a little smaller between the perfume flowers and the non-perfume flowers.
so like that rate of evolution in the sterile organs isn't necessarily positive selection, it's just.
It's just like lack of selection potentially. In fact, the rates are decoupled in perfume flowers versus non-perfume flowers. So basically in non-perfume flowers, we see that there's this tighter correlation, so this dark blue line or this turquoise line between the two sets of structures, essentially like they're evolving together, whereas in perfume flowers, they're not evolving together.
Yeah, just 2 brief questions. First of all, could I hear a favorite memory of yours just working on this entire project or working with the team? Second question, are there any future implications, considerations, or research that you would like to see in the future?
Yeah, I'll answer the second question first. So the future work, I think this RNA-seq data set has a lot, like we kind of only scratched the surface of it. I was just showing sort of the easiest pattern to talk about. I think it would be a really cool thing to follow up with genetic functional validation. And also I think expanding upon the first chapter, the morphological data set, just looking across all orchids would be really cool. As for the first question, the most fun part of this, I mean, definitely it was doing field work. I love being out there. I was really distracted by a lot of things.
Words.
Yeah.
Sorry, going back to like isolation, so does temporal as well as spatial isolation, does that fall under mechanical, or how does that work?
By temporal, do you mean like blooming at different times of the year? Or-
Yeah.
Yeah, I think that would fall more in ethological, or I think that's a separate category. But yeah, there are potential examples of that too. Santiago collected sort of this year-long data set of phenology, and it seems like there are some genera where there's like a peak during the wet season and a peak during the dry season. So yeah, that might be another mechanism. And in addition, there might be temporal in the sense of production of scent some bees are more active earlier in the morning than later in the morning, so.
You have a question online about how you determine the axes of flower morphological space and whether or not they're generalizable to flowers beyond orchids that share pollinators with them.
Yeah, so orchids have a really conserved floral morphology across all of them. The petals and the sepals might be generalizable across monocots because all of the monocots share that organization, but the reproductive structure is sort of an innovation or just like a derived trait specific to the orchid. So that would be hard to characterize outside of this group. Okay, any more questions?
Sorry, one last one.
Yeah.
In the PCA, your numbers landed around the 50% category, most of them were there, and I think there was one of them that was lower, but so is there any what's the most important thing here?
What's the most sorry, so what do you mean by the most important?
So the numbers of this are similar from like petal sepals and reproductive structures. So I'm trying to understand maybe what are some strong driving factors?
Yeah, oh, I see. Well, one of the strong driving factors might actually be taxonomy. So PC2 for both of them, that explains a large proportion of the data set actually corresponds to taxonomic organization, or two of the largest groups in this data set, which are not perfume flowers, they separate out on PC2, like the Oncididae and the Maxillarinea. They separate very clearly on the second axis, but the first axis doesn't appear to be phylogenetic or taxonomically structured at all. Okay, any other questions before I start the second part of the acknowledgements? Okay, so first I would like to thank the Ramirez Lab for providing a really wonderful place for me to work for the last 6 years. Ramirez Lab members, current lab members and old lab members. In particular, I'd like to thank Fernanda for doing a lot of the groundwork, a lot of what the foundation upon which I've built upon for my research. I'd like to thank Mika for teaching me how to do GCMS or do the floral volatile stuff in like the first quarter I was here before lockdown started. And I'd really like to thank Kathy for helping me with a lot of the transcriptomic analyses and helping me learn how to be efficient with bioinformatics. And of course, I'd like to thank Santiago. So I really like this picture that I took. I was actually photographing like the plants here, and Santiago was just there and he said afterwards, like, I didn't know what to do, so I just smiled, and it's just a really, really great picture. But yeah, thank you, Santiago, for allowing me to kind of do whatever I wanted to do and supporting my really meandering path through the PhD process. I'd like to thank my friends and family for all of the support along the way. It's been a journey these last 6 years. So thank you everyone who I've shared a meal with, who's gone out looking for plants with me, maybe trespassing slightly. I'd like to thank all the friends who I've met in the field, especially the folks at Lancaster who helped me with just getting settled in. I'd like to thank Rosa, who hosted me when I was working at Lancaster, and for like cooking delicious meals and packing little lunches for me, which is so sweet. And also, I'd like to thank my cohort. I really love this picture. It's so awkward. Like, it's so awkward. I love that I'm just standing here to the side. Yeah, we really started in 2019 and are ending in 2025. So truly, truly a time. And I'd like to thank the organisms for that really inspired my love of natural history, that have really motivated my research and made me decide to pursue a career in evolutionary biology. This is a common point of confusion for people. I don't study birds. At the field station, people were confused about that. Yeah. I'd like to thank my partner for experiencing all these things with me. We don't have fun. And finally, I would like to thank all the plants plants. These flowers are truly incredible and have really inspired me to do all the work. So with that, and oh yeah, and I'd like to thank all of you guys for coming to my exit seminar. So thank you.